so multiplication. So there are 6 genes. Okay. This has some of it. Oh, actually, all right, we'll go back to this diagram here. Don't worry. So there are 6 genes. We said it was negative sRNA, so 3' is going to be right here. And this is the order of the genes. We have N. Just from memory. I think it's N, HA, no, N, P, N, N, HA, FH, and L. 5'. This is H. I'm starting to get confused. That's H, not HA. This is the origins. And I'm going to put a short word that describes the function of each. So this is nucleocapsid. Phosphoprotein. Matrix protein. Fusion protein. Hemagglutinin. Polymerase. So, what I forgot over here, that there's these other surface proteins. I will represent them with a ball and a stick. This is F for fusion. Okay. So you can see on here, there should be 6, 1, 2, 3, 4, 5, and then 6. 6 proteins. You have N. What is it? L is a virion associated polymerase. So that was going to be found in your virion capsid. These two are also on your virion itself, so you can just see from this picture right here. Yeah. So the H allows the virion to bind to stearic acid, basically bind to your cells, and then the F allows the virus to enter your cell. It fuses the membrane of the virion, and then it just allows the whole thing to enter your cell. So that's why it's called fusion protein. Matrix just helps make the round shape, make that shell around the nucleocapsid protein in the DNA. Nucleocapsid protein, that's just so that the RNA can bind to something. So like, I guess human chromosomes, this DNA wrapped around histones, this one would be RNA wrapped around, the nucleocapsid protein helps, it's like a structure that helps give some form to the RNA. And that's the RNA bind to something so it can condense like that. Let's see, and then the phosphoprotein. I don't actually have that. 1, 2, 3, 4, 5. The phosphoprotein. So, right, phosphoprotein is a bit complicated here. So P codes for self and 2 other things. So P codes for P, V, and C. And the thing about, so P, you get the P protein from P gene, just normal transcription. But what's weird about V and C is that they are taken from the same open reading frame of your P gene, but it's slightly different. So with C, what happens is you have a start codon. You have like 2 start codons for your open reading frame, for your P open reading frame gene. One where if you start at the original AUG start site, you get P, but somewhere along the open reading frame, there's actually another start codon, and if you start transcription there, then you get your C product. The terminal sites are the same. It's just the place where along the open reading frame you start your transcription. And then V is actually made when there's an insertion of G into mRNA of the P gene mRNA transcript, and then when you when you translate that, then you get the V protein. So did I have a picture of this? Yes, I do. So we'll go. I'll draw a picture once and I'll draw it again. So 7-methyl G. So what I was trying to illustrate, what Dr. Samuels was trying to illustrate. This is open reading frame, ORF 3'N poly A tail. Okay, so this is your mRNA, your capped mRNA for your P gene. So here is, here's your AUG start site, and then this is your stop. So in black, translate that region in black, you get P protein. So P protein. But I'm going to use green for this. This is the second AUG start site. And guess what? If you start here instead of here for translation, then you code for C. Right, so you start from here, stop there, you get P. If you don't do that, you start from here instead and stop here, then you get C. And a really weird one, which is V. I did not have a picture of V. So I'll put star here by using different start site can get different protein. I remember in MCB 101B, or even like just like introductory genetics and introbio, they illustrate this concept with eukaryotic. It was like, what was it? Some weird RNA processing thing where they were saying like, oh, in high school you learn, if you're like an AP bio, you learn that one gene equals one protein. And then when you come here, they say, not really. It's like one gene can have 2 or 3 proteins. Same thing here. Okay, so here's a picture. So I'm going to try and illustrate how V is made. And we'll have this picture again later. So this is your P. This is your AUG. And your stop. Guess what? I'm going to use red for this one. If you start from the same AUG start site, and then you throw in So plus G here means you add in only one guanosine nucleotide. Back to your stuff. So what's this red? Codes for V. So what's cool about V is that literally you add in one extra nucleotide, guanosine nucleotide, and you get this, instead of a frame, I guess like by making a new frame, right? It's sort of like having a frame shift mutation where you use the idea of like it would terminate a protein early, but in the case of this V protein, you actually need the addition of that extra nucleotide to code for V. Now I remember someone in class asked Dr. Samuels, she was really confused about why is it G, why is it G, why is it G? You know, and why is it not A or U or C? And Dr. Samuels just said, because of the biochemistry of the RNA, having G is just, it's more stable. There are other viruses where they do the same thing, but they don't use G, they use A or C or U. So just that G is just something you have to memorize, I guess. Yeah, so. To make V, add one extra nucleotide to extend reading. This process is also called a really concise way. A really concise name for this is just pseudo template editing. And it's pseudo template because it's a fake template. This isn't actually really something encoded by original. This V is not really encoded by original genome of Paramyxoviridae. But by adding in a G, you make a fake template, so to speak, and you get a new gene output. So that's that. Okay. So we went into this whole picture. Why? Because we gotta link it back to multiplication. There are 6 genes that Paramyxoviridae needs to worry about. N, P, M, F, H, and F. And, but in reality, P codes for 2 other proteins in addition to itself. And that's shown here. But in the replication of the genome, you don't have to worry about coding for C or V because they are innately encoded by P. I'll take a moment, copy this down you need. It's probably the prettiest my notes I've ever seen. Okay, so multiplication. What's the next main bullet point? Well, I have it right. Where is it? Right here. So general steps. Very similar to the other viruses that we studied. You gotta attach, then you gotta penetrate, uncoat, transcribe, replicate. It doesn't matter if your virus can turn people into zombies. If you can't attach a freaking cell of the human host, you're done. Can't do anything. You gotta also be able to get inside. Your virus can turn people into zombies, and it can even attach, but it doesn't penetrate the cell. Not gonna do anything. If it's not able to uncoat, dissolve this whole round thing and expose the RNA for transcription and so forth. Can't do anything. If you can't transcribe, you can't do anything. You can't replicate your genome. You can't do anything. Okay, so the attachment is very important. Without the attachment, you can't do anything. So again, if you're trying to make a zombie virus, make sure you, at the very least, make sure the virus can attach to your human host cell. Alright, so continuing on. So attach. So specifics of attachment is that the H protein binds to sialic acid, binds to sialic acid. So when I talk about the attachment, it's basically I'll be talking about a virus that has all the different properties that all the different Paramyxoviridae can have. But in reality, certain Paramyxoviridae can bind to sialic acid, but none of the other receptors, and other Paramyxoviridae, like measles, can bind to certain things, but not others. So let's just assume that this is a universal Paramyxoviridae. Okay. It's both measles and mumps and rubella and parainfluenza and Sendai virus and Newcastle virus. You get the idea. Essentially what I'm trying to say is that with the specific species of Paramyxoviridae, we'll use specific receptors to attach. They won't use all the above, or all the ones I'll list here. So, sialic acid. So in addition to sialic acid, I thought I had heard someone coming into this room. I was kind of scared. All right. So in addition to sialic acid, you can bind the H also bind these receptors. CD46, CD155, where's 150? CD150 SLAM. And then nectin for it. Okay. So if we look at a virus like measles, measles can bind to, so CD46, no wild type Paramyxoviridae has this. Wild type no have. But vaccines. But what's important is that when we make vaccines for something like measles, mumps, and rubella, we have different lab strain of measles or mumps, rubella, whatever Paramyxoviridae that can bind CD46. So that's why CD46 is important. But the wild type of something like measles, it binds to these. So CD150 SLAM is found on white blood cells. And this is found on epithelial cells. Okay. So why these? So why is this one important? See, this is part of the reason why measles is so virulent. There's a word for it. This is what causes measles morbidity, and it's because this is found on white blood cells. It means that measles can get into white blood cells. And mess up your white blood cells. If you can mess up your white blood cells, then that means bad things for your immunity, so you get really sick. Nectin-4 is just found, epithelial cells in the respiratory tract. So if you breathe in aerosolized Paramyxoviridae, they can start from your lungs via this Nectin-4. So those are details of attachment. Then there's penetration. Here you use F protein to fuse. F is activated following binding. Basically, F changes its conformation at cell membrane. And the end result is membranes fuse. So you look on page 179 of the textbook. There's a neat little picture of your F protein. You can look in the textbook for that. I'm not going to that. Basically, it makes sense that F gets activated after it attaches. And I'm guessing this is like a concept that was illustrated in MCD 132, where Dr. Mahan says that bacteria, they have like a lot of virus factors, but some of them are only activated at certain times when they're in certain places, because you don't want those virus factors being activated too early. Because they're activated too early, then the cell can have an appropriate response in time. The analogy he gave is like launching an ICBM, so intercontinental ballistic missile. If you launch that, when countries launch those types of missiles, certain things will happen only at certain times, because it's like you wouldn't want the ICBM to explode too early, as in explode over your own country, because then you kill yourself. Okay, so you don't want to do that. You don't want it to explode over the opposing country, because then you might not do that much damage. You don't want to explode midway to the country, because then the opposing country can detect that you're launching ICBMs and it'll prep its defenses, its anti-ICBM defenses. What you do want to do is you want to explode and detonate its nuclear payload when it's flying inside a country, inside of its city. So that long analogy is the same thing with bacteria, same thing with viruses. You don't want membrane fusion happening when it's not even next to a host cell. That'd be a waste of F protein. So you wait until you attach using H first, and then you activate your F. And when F is activated, it's a protein. It changes the shape a bit, and then diffuses the virus into the host cell. Okay, so I'm gonna erase this. Okay, so after penetration of the virus into the cell, you uncoat and do partial encoding. That's all Dr. Sari said about that. I'm guessing you can, I think it was chapter 4 that it might talk about encoding and so forth. So after you uncoat, now the genome is in the cytoplasm, you can transcribe it. So for transcription, there's only one start site for an entire genome, just like Rhabdoviridae. You get attenuation into gene junction. So this is where, if you recall, it's where the polymerase, separating the 6 genes of Paramyxoviridae, or these tiny, I think it's 3 nucleotide sequences where the polymerase can literally just fall off at those areas, so that you get more mRNA product of the genes that you need most. For example, nucleocapsid protein, you're going to need more of that than your polymerase, which is an enzyme. You don't need as much enzyme as you do structural protein. So attenuation happens, and again, similar like Rhabdoviridae. And then polymerase stuttering, just like Rhabdoviridae. You get those stuttering. They're poly U nucleotides in the native SS RNA genome of Paramyxoviridae, in order for the polymerase to stutter and slip on those poly U's and then make the poly A tail. It's just like Rhabdoviridae. You can probably already start to see like why in the textbook they group Paramyxoviridae and Rhabdoviridae together. Okay. Remember, because they only have one start site, I'm using Paramyxoviridae, just like Rhabdoviridae, you shoot radiation at it, just like in a discussion section paper, you're going to you get the same results. So, transcribe. And finally, of course, oh, there's a picture. I'll end this list with replication. So after transcription, you get replicate genome. One important thing is that most viral protein made during secondary transcription. So just like Paramyxoviridae, you have a native ssRNA. It goes in, you transcribe it, and then you replicate it a bit. And then, so that's like, that consists of the whole first transcription replication event. So it's the first time it does it. And then it takes the newly replicated genome, so it has many more copies of its native ssRNA, and then it does transcription on those. So instead of you transcribing proteins from just one native ssRNA, and making some X amount of mRNA from that one transcript, now you have, or one genome, now you have a bunch of native ssRNA, and each of those will have a bunch of mRNAs, and from those mRNAs you have even more protein made. So most of the viral protein that is made, most of the viral protein made by Paramyxoviridae happens during the second transcription event. The picture that I want to show next is, so the picture that I want to show or draw was for step 4. And again, I think if you look back in the Paramyxoviridae video, I had this picture in there somewhere, but just in case, for good reveal. Junction, C here. Okay. So you see, this whole region is the N gene, and then you have this tiny intergenic junction, and the rest of this is the P, and then the M, and then the F, the H, and the F. Okay. So this is your genome. You see the poly U, U sub 8. So when you're making the mRNA transcript, obviously you need the 7-methyl G, Y prime, copy your N, and finally when you get to here, you get the complement of that, and then you get your poly A tail, 150 A's. So you see U, A, U, complements of each other, just like normal. Remember this right here on a preliminary sketch right here, it stutters, it slips. It's like it reads, it reads a bunch of U's, and it's like, oh, dang, maybe that's wrong. We'll go backwards and read again, or we'll go backwards and read again, just in case. And we always get poly A tail, so that's how poly A tail is made. And when it's transcribing, it does all this stuttering something, gets to the intergene junction, polymerase falls off, it attenuates. And that attenuation leads to you having an mRNA of just the M. Now, of course, sometimes the polymerase can move on and continue with the P, so you get N and your P. So 7-methyl G. And then you get A, G, G, A, blah, 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 3 prime, so you can continue making, you get to intergene junction. You don't transcribe the intergene junction, but the polymerase can go read through the poly U area, go through here without falling off, and then it can continue transcribing the P and so forth. Or it can stop right here, it can fall off, and all you get is just a bunch of that. And again, it uses this strategy so I can transcribe, so I can make more of the protein that it needs more of. Again, you need more N than you do L. You need more of the structural protein than you do polymerase.